next up is Nat Kendal Freedman. I can barely see, so I'm sorry. I missed letters there.
It's toss-up whether people add or remove letters. I've been told I should bring my own stopwatch. All right, so just before I get started, I want to take a second to thank my supervisor for his support of this project, even though it's essentially completely unrelated to the work we've been doing together. So a huge shout out to Brian Ingalls for that. And also to Thomas Hillen and Jay Newby for all their help on this project when I was at the U of A. So with that, we can go ahead and get started. And we're going to be doing about a 180 from the last talk because machine learning tends to give really good results without a huge amount of understanding of what happened in the middle. Whereas what I'm going to be doing is going to give very wacky looking results, but we will understand exactly why that's happening. So that should be a fun difference. And the other thing I should say with that is that I am first and foremost a mathematician. So we're not going to be looking at quantitative results. We're going to be looking at qualitative results. We're going to be trying to see if we can find some missing pieces of the story and put them together. And then we'll worry about moving on and making it actually accurate. But the first thing is to try to look at what is and what isn't appearing in our models. And so to do this, what we'll be looking at is CAR T cell therapy, and in particular, looking at the role of T regulatory cells on relapse. So to start with, as I just said, I'm not a biologist. We're going to begin with the biology. So for those of you who don't know, chimeric antigen receptor, or CAR T cell therapy, is a very common type of immunotherapy for things like blood cancers, lymphomas, and more recently, we've been looking into using it with things even like melanomas, things that are metastatic. And so the way this works is that it's a very specialized treatment to each patient. You draw blood from the patient, isolate their T cells, and then you engineer them to present receptors specific to the antigens on the patient's cancerous cells. So in the case, for instance, of B cell cancer, B cells tend to present an antigen called CD19. And so we will essentially train the cells to present a receptor that will specifically look for CD19 and be able to detect that and therefore more accurately target cancer cells that would otherwise be missed. And this is why it's particularly effective because it's from the body's own cells, so the body isn't rejecting them in most cases. And the other great thing we can do is we can grow these CAR T cells really, really quickly in the lab compared to what the body can do, so we can hit you with a very large dose of them. But of course, as with anything like that, there are toxic effects to treatment, right? Having too much of a good thing is still going to be too much. So it's just like if you drink 20 gallons of orange juice at once, it's going to be bad for you. If you get this many immune cells at once, it's going to be bad for you. So what we do is we give a round of chemotherapy first, not with the intent of killing off the disease, but with lowering the burden enough that you can tolerate the necessary dose of treatment. All right, so this is the sort of therapy that we'll be thinking about, so just good to keep in the back of your minds. And with that, let's look at some of the current models. So Leon, Triana, and Sabir have a great model. They look at a lot of the effects of immature cells in the bone marrow, things that have yet to differentiate, which is great because a lot of blood cancers actually start in the bone marrow. But what they also have is they have a simplification of their model for a shorter time frame. And that's what I have here. And you'll notice that it has very few terms, which is great when you want to be able to understand exactly what your model is doing. And it also has results that are very consistent with other models. And so just to take a look at what they have, we have, for instance, here, a simulation of where they predict relapse. And in particular, what they do is they predict relapse as a consequence of prey-predator dynamics. So you'll see, with my lack of pointer, hopefully, that after the leukemia relapses, that red line, there's a little green spike. And that's the CAR T cells coming back in their model once the leukemia comes back. But there's a slight problem with this. And the problem with this is that in clinical trials, CAR T cells persist for maybe 2 or 3 months. And they certainly don't persist for over a year. And that means that we're missing some sort of fundamental mechanism in our model. And that means we need to look back and see what we might be missing. And moreover, we can actually see this in the original model. Can we go back? Can. So if you'll notice, there's exactly one death term for the cancer, and that's from the CAR T cells. And what that means is that the CAR T cells cannot go extinct if the cancer is still present, because you get a prey predator model with exponential growth. And so you will always get these sort of cycling behaviors that we've seen previously with those like wolf and moose diagrams. And so that's another good indication that we definitely need to be adding something to this model. And the other thing you might notice is that the B cells are not having a great time. Everything's killing them, and they're really not going to be growing fast enough to matter. And this is actually interesting as well, because this is a great sign of when CAR T cell therapy is working. The best predictor of it is actually if you have B cell aplasia, which just means you have no detectable B cells, which is somewhat unfortunate, of course, but a lot better than having cancerous B cells around. So we'll be omitting that in our model because they essentially die right at the start. As you might notice, the B cells go straight to 0 and stay there throughout. All right, so throughout the conference, we've been hearing a lot about phenotypic plasticity or changing of the tumors. But we haven't talked a lot about the immune cells changing, and it turns out they can. And so to give a kind of example of when this has been applied to a model really successfully, I get to plug for my old supervisor, Thomas Hillen, and one of his students, Adam Rhodes. And they talk about this immune-mediated theory of metastasis. And they argue that the key to it is this inclusion of what they call pro-tumor immune effects. And this is when, instead of the immune system doing its job and being helpful and killing the cancer cells, instead, for whatever reason, it gets corrupted and helps the cancer cells. And by adding these into their model, they're able to explain tumor dormancy. So this is when a tumor just lays dormant for unknown reasons for a really long time before suddenly coming back. They're able to explain metastatic blowup, which is where you have a primary tumor, it's removed via surgery, and then nearly overnight, a secondary tumor pops up. Previous models weren't able to explain this accurately. And likewise, they were able to explain metastasis to sites of injury. So if you already have cancer and then you break your arm, and then suddenly you end up with a metastasis in your arm. And this is partly because that is a site with a lot of inflammation, and inflammation means immune response. And so it's something to do with the fact that there's more immune cells there that is being counterproductive and somehow helping the cancer. And so this is a great justification to say maybe we should look at this as well. But of course, we're going to be looking at a slightly different case. So we're going to be looking at T regulatory cells. And T regulatory cells are just the part of the immune system that are responsible for saying, okay, guys, we've done our job. You can go home now. We're done. They're kind of the opposite of the effector cells in that sense. And normally they'll be maybe around 5 to 10% of your total cells, because right, most of the time you want your immune system to be doing stuff. But extremely counterintuitively, in cancer microenvironments, they can be up to 20 to 30% of your T cells, which is the exact opposite of what you'd think and the exact opposite of what you'd want. And so the question is, why? And the first thing we look at is this plasticity. And it turns out that T regulatory cells don't actually have to develop originally from other as T regulatory cells. They can also be converted from other T cells. And the next question, of course, then is, well, what is the pathway that could lead to this conversion? And the answer is that there is this cytokine called transforming growth factor beta, or TGF beta, and it's secreted by cancerous cells. And sure enough, it promotes the conversion of T cells into T regulatory cells. And so our hypothesis here is that if we add in the effects of T regulatory cells and TGF beta that's being secreted by cancer, maybe we can reproduce the phenomenon where CAR T cells always die out, regardless of whether the cancer does. Maybe this is kind of the missing link. And so that's what we're going to do. We're going to add these to the original model that we showed before and see what happens. So we have now 4 equations, as before, CAR T cells and cancer, but now we've added on T regulatory cells and TGF beta. And we will also might note that we've replaced the exponential growth term with a logistic growth term. And this is partly because if you only have those CAR T cells as the killing factor, it's mathematically impossible to end up with the CAR T cells dying out. And what we don't have in this model is regular effector T cells, the ones that are killing the cancer. And there will still be some of those around. And similar effects like this can be more simply accounted for by including some sort of carrying capacity and saying, okay, growth will slow down at some point. It's not always going to skyrocket. And beyond that, the growth is going to be such that it's going to be a high enough carrying capacity that realistically it's unlimited as far as the human body cares. And so the new terms we've added here are inactivation, of the T regulatory cells towards CAR T cells, as well as conversion by interactions with the TGF-beta. And you'll notice that essentially everything is the law of mass action, because we're really just going for the simplest thing to see if we just add a little bit to this model, can we really change the dynamics that it predicts? So let's look at what we get. So the first equilibrium we often want to check is the equilibrium where everything dies. The cancer is cured and the CAR T cells also die. And the good news is that this is stable with respect to the CAR T cell population. So even a small fluctuation, it will allow it to die out, but it is unstable with respect to cancer. And that means just if you get a little more cancer afterwards, it will re-go. So this is exactly what we'd expect. And of course, this is where I'll have to remind you that, of course, it's not realistic for CAR T cells to take 150 days to die out when the cancer is dead in about 2. But again, it's about the asymptotics that we care about rather than the exact number. And likewise here, we have relapse in the absence of CAR T. This is what we were hoping to be able to find. And indeed, not only can we reproduce this, but we can find a condition for it to be stable. So in particular, we need the stimulation to be sufficiently small compared to the inactivation and the secretion of TGF-beta. And so what we see here is, again, a little bit of a janky plot, but the cancer cells in orange, they die out, and then they start coming back. But the CAR T cells, once they decline, they stay dead. And this is something that the other models so far haven't been able to reproduce. And it's a great piece of evidence that these T regulatory cells are what we should be looking at. And finally, we need to see if our model ever does have coexistence. And the answer is it does, because it still is going to be fundamentally a lot of prey-predator dynamics here. But what we find here is that we can find a condition that is necessary for instability. And it's these same 3 parameters, but opposite. The inactivation and the secretion have to be sufficiently small compared to the stimulation. And that's interesting because now we can get a range for our parameters. But I will also point out, because if you're me and you look at this first diagram, your question is that convergent or not? And if you run it for about, 10, 100,000 years, it will eventually converge. And this is where I say, this isn't going to be biologically accurate, but in the limit, in long time, it works out. And so just to summarize the exact conditions for any of the mathematicians out there that care, these would be the conditions we would need to get a realistic model. And I will note these are non-dimensionalized parameters, so they won't correspond one-to-one with what you saw in the model earlier, but they still represent the same biological phenomena. So the CAR T cell extinction, to be stable, omega, the inactivation, has to be greater than the stimulation minus the secretion minus 1, and that's just like the regular death term of CAR T cells. That's where the 1 comes from. And then for instability of persistence, it's necessary that omega is smaller than this rho minus sigma plus 1. And in addition, we do require that the cancer population is no more than about half its carrying capacity. And this is just because if it is so great, the CAR T cells are going to be stimulated enough that it will overcome these negative terms we've added in. And it is a realistic assumption to have, because generally with CAR T cell therapy, we either see a strong response in which the tumor population really does plummet, or very little response and it doesn't work at all, in which case this model isn't really accurate anyway. And so with that, I want to talk about the current work we're doing. And so like I've said throughout this, my model is not going to be used to predict patient outcomes anytime soon. But what we can do now that we have a justification that this actually works is move into a model that is experimentally validated. And the model I've been working with actually happens to be partly made by someone here. Ivana and one of her students, Catherine Owens, made a great model of CAR T cell therapy that I've been working with. If anyone's interested in the results I have from that, I'd be happy to talk about it. It's just far too complicated to do in about 10 minutes. And so we're going to be using this to investigate the effect on patient outcomes, to confirm that we see the same sort of overall dynamics, even though we're no longer able to analyze the model mathematically speaking. And the other fun thing is that after the talk today earlier about sex differences, I was doing a little reading, and it turns out that T regulatory cell populations are different in men and women. And in particular, they tend to be higher in women, higher in men than in women, meaning that this could actually explain a large portion of what we talked about. Because if there's more T regulatory cells, your therapies are going to be less effective. because more of your cells that you're inputting into the body are going to be converted and then end up having a negative effect. So that's something interesting and something we'll be talking about later today if anyone wants to join us. And with that, here's a list of references in case anyone wants to actually know about the biology that I only vaguely understand. I can point you to the right one. And to thank you to the funding from NSERC, U of A, and Waterloo.
Right, very, very interesting dynamics. So I'm curious about the instability and how this might relate to model structures. You showed that there's lots of oscillations with the CTL, right? Or with the T cells. And yeah, so I've dealt with similar immune system models in terms of viral infection. You see the same kind of things. And I think in your immune response stimulation term, it's proportional both to the number of T cells and the amount of antigen.
Yes.
So if you change that formulation and you put a saturation term in,
exactly,
then it becomes stable. Or even,
it can be, depending.
Yeah, it becomes much more stable, much better behaved. In fact, you cannot make it proportional to the amount of antigen, to the amount of T cells at all, and then it's very so it really, and we really don't know biologically how to describe this, right?
There's some good evidence that stimulation occurs through interactions with the cells presenting the antigens. So it is at least somewhat of the right flavor term.
Some of the biology says that when a CTL recognizes antigen on an antigen presenting cell, it triggers a program in the CTL, which is then completely antigen independent. So there's a program of 8 divisions or something, and then it goes into effectors and they do their thing and then go into memory. And so there are all sorts of argument with that. So it might be worth looking into this a bit more detail and see how that affects the
Yeah, absolutely. It's definitely one of those terms that needs to be looked at because, for instance, there's no immune cell exhaustion here.
Right.
There's no regular T cells that are competing for space.
Exactly.
And that's all things that are included in the other model we've been looking at, which also makes it exactly why we can't analyze it well.
Yeah. No, it's very interesting dynamics.
Thank you.